can see it now.
I hope everybody can see this.
Yeah, sir, it's visible.
Can you see this, ma'am? Yep. Going with the vaccine development for COVID, currently speaking, there are nearly around 83 companies which have registered their vaccine to bring it into production. Out of these 83, 77 companies have been given clearance for clinical trials globally.
Right.
So with respect to this, if we see the first one to come out is that is the first of its kind, to be frank with, an mRNA based vaccine, which is restricted so far because mRNA based vaccines have its own side effects, and there are many organizations against DNA based vaccines. So these kinds of vaccines have got a high chances now because of this COVID situation, because there is a high need for this vaccine for COVID, as it's been spread globally, and we have this is the overall procedure how a vaccine is being developed. So if we see before getting into production and marketing, we have to go for several phases of vaccine production. So basically splitting it into two categories. One will be preclinical and other one will be clinical. So
Right.
if you see clearly, the preclinical phase where we go for vaccines, this will be involving all the lab researches with a plant, even whether it's a plant extract or a drug molecule design, from the
Right.
computational biology and from the dry labs to wet lab. And from wet lab, once if you are affirmative with the target results, then these will be carried on to animal testing.
Right.
So in animal testing, we will be studying the side effects and other effects of these drugs with respect to that animal which has the most similarity towards a human physiology.
Human.
So mostly preferred one are the mice. So mice are the experimental animals which we use for testing all the drugs. And from that, then we enter into a phase called as clinical testing. So
Right.
clinical testing is done in nearly around 4 stages. One is phase 1, phase 2, phase 3, phase 4. The
Right.
phase 1, we will be directly targeting upon the patients who are infected, and with their consent, with the involvement of the patient and advising about the risks they will be facing, and having their consent, we'll be proceeding with the trial. So
Right.
here we will be studying how the disease is being addressed. By understanding that, we will be able to come to conclusions whether our drug will be having a mixed population as well as a huge population, where we will be concentrating on the side effects and other adverse effects of the drugs, because all human physiology are not the same. It
Right.
differs from place to place and race to race.
Right.
So we have to go through and analyze the drug's effect in each and every condition, how the drug functions, whether is there any deviation or is there any adverse effects because of this drug. Because a person who might have some particular disease can be a normal person, he might be a cardiac person, he might be a diabetic person. So we cannot be sure that the drug what we deliver can be the same for all these two together. So we have to analyze all these things, so it will be taken to phase 2. Once we have got the results for phase 2, we'll be going to phase 3. So the phase 3 will be a even
Yeah.
more huge population. Based on the long-term effects, we will be also analyzing over how this drug's impact is there. So depending on this, we'll be formulating the results. So finally, it will be going for phase 4 or post-marketing surveillance. So post-marketing surveillance is a common kind of a drug trial that has been done by all the pharmaceutical companies to analyze the efficacy of the drug. So the drug is efficient, they're good, but
Sir,
the efficacy
your
means
voice is
how
not clear.
long and how competitive and how efficient it has been over a long period of time. So this, ma'am, my voice, is it not clear for everybody, ma'am?
Yeah, yeah,
Is
yeah.
it clear now?
Carry on, sir.
Hello?
It's better now. It's better. It's
Yeah.
better. Please carry on.
So Yeah, so these are the phases that is involved involved during drug development. So this, if we see how long it will take, the actual period, unless it is a crisis, it will take
Right.
minimum of 10 years for the preclinical and the phase 1 clinical study. So
My God.
preclinical and phase 1 clinical period is 1 to 10 years, and then phase 2 and phase 3 proof of concept is phase 2 proof of concept. This will take around 2 to 3 years, and
Right.
then another 2 to 4 years for phase 3, and finally into the market it is the post-marketing surveillance, that is around one year of a study. So overall, if we just see the amount of years invested in a drug to come into the market, it takes nearly around 20 years.
20
But
years.
with respect to COVID, in crisis like this, there is a need for an, yeah, there is minimum of 15 to 20 years that is being needed for a drug to come into the market. This from bench to bedside, translational medicine, a translational research, which means from bench to bedside. Bench means all the wet lab, in vivo, in vitro studies that we do, and all these clinical trial practices, and finally this reaches our bedside. So this process will take minimum drug without adverse effects and all. Any drug if we take, there will be side effects. Whether the side effects is minimal or is it going to cause a major issue for that physiology, that matters a lot when it comes to this particular kind of studies.
Right.
So it will take nearly around 15 to 20 years. But when
15 to
we
20
see
years. But do you
at the
foresee
current
for COVID?
situation, there is a need for an immediate drug.
Right.
So now, yes, ma'am, that's what. For COVID, what is happening is, I would say for COVID, people have recommended for an early release of drug, early approvals of drug, even if we, that's what. The current slide, what I show is the DCGI has taken a fast
Right.
track approach for these drugs. So it has given a high priority for approval of all these
Clinical
researchers, all these foreign companies and clinical practices, and all the studies, toxicology study, everything they've given immediate. So this will be happening parallelly.
Okay.
It is not that they will be bypassing all these researches. The researches will be happening parallelly. So if one in vivo study and the side effect issues is being handling by a lab, and similarly a phase 1 study with a limited population and a limited time span will be run parallelly, and to see whether there are any adverse effects. So this phase will come only after addressing that in the lab. Without
Right.
addressing it, they won't bring it to phase 1. So
Right.
after addressing it, other side effects will be studied in the lab as well as in the field,
Right.
clinical practices. So this will go on simultaneously, parallelly, to cut down the number of years. So that is how we will be able to provide the results. But
So by
with this, we cannot say
adopting
that
parallel
this will be the
approach,
most
we can cut
efficient
down
way.
on time.
Yeah, adopting parallel approach, we can cut down in time, but the second step involves the result of the previous step.
Right.
So simultaneously, to have the results side by side in order to get the steps done. So,
So it
but if
requires
we see here, ma'am,
collaborative efforts,
sorry,
Rahul.
exactly, collaborative, and the timing is very much important, because if the if one work is being withheld, the simultaneously the corresponding works would not take place. So that
Absolutely.
kind of an issue will be there if the timing is being missed.
Right.
So the ethical negatives of, we call this ethical negatives because there is no other go. This is a crisis and we have to
This is
handle the
a crisis
situation.
situation.
There is a high, yeah. So there is a high chance of false positives.
Right.
So here we have reduced the sample size. So since we have reduced the sample size, there are high chances of getting false
False
positives.
results.
If
False
you see
results.
what is false positives, the results might show it is positive, but we may not know whether it is technically right, because we will be, for example, we can say that the Chinese kit which we are using for diagnostic nowadays,
Right.
which has been reportedly 55% accuracy, 60% accuracy. These are because of the false positives. So there is
Right.
a high chances of false positives with these tests, and the surface analysis is only possible. So you cannot do in-depth analysis. You cannot categorize the patients